So overview of our talk on the developing brain. We're going to first look at the brain, and then we're going to look at parental and environmental influences on the brain, media influences on the brain, and spiritual influences on the brain. The brain has 100 billion neurons, approximately 100 billion neurons or nerve cells, over 1 trillion supporting cells, and each brain cell can have up to 10,000 connections to other brain cells, makes it highly interconnected, up to 40 quadrillion interconnections. And these interconnections are really critical to have a smart network circuitry in your brain. Albert Einstein donated his brain to science, and they biopsied his brain, and he had less neurons per cubic centimeter than the average person. But he had significantly more neuron-to-neuron connections than the average person per cubic centimeter. And it's those neuron-to-neuron connections that make a complex circuitry or system where information can be processed from multiple different angles, and that is really critical, and we get that by exercising the various neural circuits. The brain is soft, kind of like a banana, inside of hard case, your skull, so a rapid acceleration, deceleration will cause the brain to bounce on your skull, causing it to bruise. We call that a concussion. And concussions, the more concussions you have, the more risk of dementia you have later. Number one reason for head injuries in America are bicycle accidents. And the recent study showed that more headers in soccer are associated with dementia when you age. So we all know about regular American football and the head injuries that are associated with dementia there. The brain weighs around 3 pounds, which is, and I'm sorry I didn't convert that to kilograms, but I think it's like 1.5 kilograms. And it's 1 to 2% of your body weight, but it uses 20% of your body's energy. The human brain is so complex that more than 50% of the genome, or the information in your genes, code for your brain. Even though it's only 2 to 3% of your body size, 50% of your DNA codes for the brain. That's how complex it is. During brain development, more than twice as many neurons are produced in fetal development than are eventually used. And during fetal development, there are times when 50,000 neurons are being produced per second. From birth to the first 8 years of life, the brain is busy killing off neurons by the millions. A child at age 8 will have millions of neurons less than that child had when that child was born. At first, this doesn't sound too good. I know some of you are thinking, I needed that extra several million neurons. But the way you conceptualize this is Michelangelo's block of marble when Michelangelo gets it, and Michelangelo's block of marble when he's done with it. When he's done, he has less marble, doesn't he? But he has a masterpiece. The brain comes into the world prepared to be acted upon by education, environment, circumstances. Neural circuits which are being fired and exercised will expand and grow more complex. Neural circuits which are not being used will be pruned back and deleted. This is normal brain development. When you understand this process, it's critical then you can introduce activities and experiences into children's brains that will influence the actual developing circuitry of that brain. A second wave of neurogenesis hits and peaks in girls at age 11 and boys at age 12, followed by several more years of neural pruning. And this is, of course, the neurons that are coming in just before puberty to help the development of secondary sexual characteristics. And these years of childhood development are vulnerable years for exploitation. If kids are exploited or exposed to various sexual deviances, they can actually have altered arousal circuits where things that normally wouldn't arouse them would contribute to arousals, particularly the paraphilias, which are things like voyeurism, frauderism, pornography addiction, and things like this during these developmental years. White matter maturation progresses from the back of the brain to the front of the brain. The last part of the brain to fully develop is the prefrontal cortex, where you reason, think, organize, plan, strategize, self-restrain. It's really the highest portion of our reasoning capacities, and that finishes around age 25. When shown faces and asked to describe what they see, the emotions, younger teens will activate their amygdala or emotion circuits. Older teens will begin activating prefrontal cortex to think about it before they answer. So parents influence the developing brain both by the heredity, the genes they pass along, and the environment in which they raise the child in. Now, there's a term called epigenetics. It came up by Conrad Waddington in 1957. He noticed that every cell of your body has the same DNA, the same chromosomes. Yet your skin cells are quite different than your bone cells, which are different than your heart cells, which are different than your brain cells, which are different than your retinal cells. But all the different cells, so different, have the same complement of DNA. So he hypothesized there must be some set of instructions sitting inside the DNA or on the DNA, telling the DNA in the different parts of your bodies which genes to turn on and which genes to turn off. And so he coined this term epi, meaning above, and genetics, the genome, epigenetic instructions. And in fact, science has now proven this is exactly true. There are epigenetics. There's instructions that sit above the DNA telling the library of information which books to open and gather the recipes out of to produce those proteins and which ones to keep closed. And these epigenetic modifications we're going to explore how those get passed along down through generations, things that happen in intrauterine environment that alter those expressions, and then early childhood environment. So as we go into the epigenetic data, I want you to consider these two mice and consider how closely related are they. This is a mother and a child from one of its litters, a mother and a grandchild or a great-grandchild from one of its litters. Are these mice that are brothers and sisters or cousins, or how closely related are they? We will come back to it. So this is simply a diagram of chromosomes, and you remember your DNA is a double helix. In order for the information that's stored in your DNA to be expressed, this double helix, has to unzip. And the way DNA is stored, it's two sides stored together, zipped together, wrapped around histones, packed into chromatin, and packed into chromosomes. So in order to get the information out, it has to unpack, unzip, and unwind. And along that way, there's places things can do to shut that system down. So methyl, little tiny methyl groups, chemical group, if it attaches actually to the DNA helix itself, the zipper itself, it's like, just metaphorically think about getting a shirt tail caught in your zipper. You get a shirt tail caught in your zipper will not unzip. These methyl groups do that to DNA regions wherever they attach. They lock them down, and this area will not unzip anymore. So the information there is no longer being expressed. You can also modify by putting various chemical groups along the histones where the DNA is wrapping around, and that can either stimulate more expression, cause it to unwind and express, or to lock it up where it won't express. And there are other modifiers as well, including non-coding RNA. This has been ignored by biologists for years. They had no idea what it was. They never taught you about it in Biology 101. It just wasn't even talked about. But now we know that the non-coding RNA means not making protein, so it's not coding in the sequence of protein production, will actually splice genetic material together, modifies gene expression, so it actually affects how the epigenetic expression, which genes are turned on and which genes are turned off. It suppresses an entire chromosome. Many of you know that women have 2 X chromosomes. Each cell only needs one of those X chromosomes to express itself. So every cell in a woman's body will randomly, individually decide to shut one of those off. And woman gets a X chromosome from mom and an X chromosome from dad, and every cell in her body randomly decides to turn one of those off. So some of her cells are expressing the X chromosome from dad, and some of the cells are expressing the X chromosome from mom. And this can contribute to the reason why women may have a slight increase autoimmune type disease, because there's subtle differences in how those chromosomes may express themselves. For instance, many of you know that color blindness is carried, or color vision is carried on the X chromosome. So if a man gets the bad gene for color vision, he only has one copy, he's colorblind. Women get 2 copies, so it's very rare for a woman to actually be colorblind, because she'd have to have 2 bad genes in order to be colorblind. But with our new technology with laser precision testing of the retina, if a woman has a colorblind father, she won't be colorblind. She looks at stuff, she'll be able to tell. But if they actually go in and look cell to cell, some of the cells in her retina will be colorblind, because some of the cells in her retina will randomly turn on dad's X chromosome, and that cell can't see color. But the ones from mom that randomly turn on, she can still see color, but maybe not as precise and as clear. So in dim light, she might have a harder time seeing color than she would have if she had 2 good genes. So it also suppresses, so the non-coding RNA suppresses that entire chromosome. It elongates telomeres, and telomeres are these, like the caps on the end of your chromosomes. Think of it as the plastic caps on the end of your shoestrings. It keeps your chromosomes organized, and you have to have certain length telomeres in order for the cells to reproduce and copy themselves, and this is a factor in aging. When the telomere is short, the cells can't copy themselves anymore, the cell's thin, it's harder to heal after an injury. This is why older people it takes much longer to heal after an injury, because their telomeres are very short and they can't produce cells as quickly. And then defense against invading or DNA sequences that are not natural to your DNA. And so these are called transposons, where some piece of DNA gets inserted into your DNA where it shouldn't be, and the non-coding RNA will come in and lock that down so it can't express itself in damaging ways. So with that in mind, looking at the epigenetics and how it can affect things, they took fruit flies and they exposed them to an antibiotic called geldenomycin. And the generation that was exposed to geldenomycin had these very weird outgrowths on their eyes. Then they let this generation that had these weird outgrowths from the exposure to the geldenomycin begin to reproduce. And for the next 13 generations, every generation subsequent had these bulging outgrowths on their eyes, even though there was no DNA sequencing change. In other words, there was no mutation in the actual DNA sequence, and generations 2 through 13 were not exposed to the drug. So this exposure to the drug altered the eyes and was passed down 13 generations because the epigenetic information was changed, and that altered how the gene was being expressed. Roundworms were fed a particular type of bacteria that caused them to exhibit a kind of dumpy appearance and loss of fluorescence, and that passed down 40 generations, even though the subsequent generations were not given this bacteria, and there was no DNA sequencing change. In other words, no mutation in the DNA. Transgenerational effects on food supply. Grandfathers who had short food supply during their adolescent period conferred greater risk of mortality onto their grandsons, and grandmothers had greater risk of mortality conferred to their granddaughters if they had a very short food supply, like during World War II, for instance, to their granddaughters. This was XY epigenetic modification based on the food supply during their adolescent period. Their genes altered, and they passed that down to their grandkids. Men who smoke before the age of 11 increase the obesity in their sons, but not their daughters, because it alters a gene expression on the Y chromosome that is passed down to their sons. Smell of fear. There's an interesting study. They took these mice, and the zero-generation mice, and they were conditioned to fear a particular smell called acetophenone, and that's because these mice have an actual single receptor that smells for this particular smell and nothing else. And so this particular gene codes for that receptor in their olfactory bulb. And when they would expose them to acetophenone, at the same time they would make a loud bang. And so every time they exposed these mice to acetophenone, they would make this loud bang and startle them and scare them, so they conditioned them to fear this smell. And that activated this gene, which caused that particular receptor in their olfactory bulb to be increasingly expressed because they became more and more sensitive to that smell as they became afraid of it. Does that make sense to everyone? Now, that's not surprising. What's interesting, though, is that generations F1 and F2, which mean their children and grandchildren, had decreased methyl groups on the gene that codes for this particular receptor in their olfactory bulb, so they had an increased expression of this particular olfactory smell, meaning that they were born afraid of this smell, having never been exposed to it. Genetic memory, perhaps, but they're already conditioned to fear it, and they were never exposed to it because of what happened to their parents. Gestational factors. Smoking while pregnant, smaller babies, increased risk of SIDS, more learning and behavior problems, increased psychotic disorders later in life. How about alcohol when pregnant? Well, we all know increased psychosis, alcohol. Did you know that if your mother drank alcohol when she was pregnant with you, it would epigenetically alter your taste buds so that if you drink alcohol, it would actually taste better to you than had she not drank when she was pregnant with you? Increases risk of mental retardation, multiple organ defects, fetal alcohol syndrome. But what about this? What about one drink of wine, say, or beer per week or less? Just one glass of wine a week or less while pregnant. That's not going to be significant, is it? Actually, that much alcohol while pregnant, shorter babies with smaller heads, more behavior problems later in life, more delinquency later in life, and more emotional problems later in life than a mother had no alcohol during pregnancy. Alters brain development. What about mothers thinking while pregnant? Does what mother thinks while pregnant affect the developing brain? Interesting study of over 4,000 mothers and their children followed for 18 years, and they found that mothers with negative, pessimistic, depressogenic type thinking patterns increased the risk of depression in their child 18 years later. And the association remained after accounting for other maternal offspring factors of depression, and they concluded in the study that negative depressogenic thinking while pregnant will increase the risk of your child being pregnant by 21%. just by negative thinking. What about if the mother's highly stressed during pregnancy? And it may not be her fault. She may. In America, we've been sending troops over to the Middle East for a long time now. So a young woman gets married and she gets pregnant and her husband gets called up in his National Guard unit and sent over to Afghanistan. Do you think she's going to be stressed? Yeah, she's done nothing wrong, but she's going to be stressed. Or how about a woman who's pregnant gets called that her mother's just been diagnosed with ovarian cancer or breast cancer? Is she going to be stressed? Lots of things cause stress. It doesn't mean you're a bad person, okay? But people get stressed. If a mother is stressed during pregnancy, the stress hormones, the glucocorticoids, cross the placental barrier, cross the developing blood-brain barrier of the child, the fetus, and alter the braking mechanism on the alarm circuitry, the amygdala, such that the children are born more prone to irritability, anxiety, mood disorder problems than had mother not been highly stressed during pregnancy. And this happens through epigenetic modification of the system that helps calm. So the babies are a little more irritable, they're less capable of calming, and they're overreactive, more anxious to environmental stimuli based on mom's stress during pregnancy. Diet of the mother during pregnancy. So how closely related are these mice? Yes, some of you already said it. These are clones or twins. These are identical mice. Identical genetically. Do they look identical to you? No. Well, there's a gene called the agouti gene. When the agouti gene is turned on, then the mice are obese, diabetic, and blonde. That's what that gene does. Obese, diabetes, and blonde. If the gene is shut off, the single gene, the mice are thin, non-diabetic, and brown. And you can see there can be partial penetrance of this gene expression. Here is you can see some brown fur, you can see some brown fur, and you can see some brown fur coming through different places. And so the glucose tolerance and the weight and the obesity, depending on how many of the genes, because every cell has its own gene in it, so how many of the cells have it on, how many of the cells turn it off, that will affect the health of the mouse. Well, what turns on the agouti gene? Because if it's on, you're obese, diabetic, and blonde. What turns it on? Bisphenol A, which is a byproduct of plastics. Bisphenol A will turn this gene on. And I put the cans on here because every can, if you eat canned food, it's lined in plastic so that the plastic protects a protective layer so the chemicals and the acids and things in food won't cause a chemical reaction with the metal. So bisphenol A turns on. Well, what turns off the agouti gene? Methylation, a methyl group. And so they took these obese, diabetic, blonde mice who had the agouti gene turned on, and they had them get pregnant, and during their pregnancy, they gave them a diet supplemented with folic acid and B12. Folic acid and B12 are methyl donors. They give lots of methyl groups over. And their children, the pups from these mothers, were born with the agouti gene turned off. They were born thin, brown, non-diabetic mice. And then they let the children have children, but their diets were not supplemented with the B12 and folic acid. And their children were born with the agouti gene turned off. And they let those have more. And so this agouti gene being turned off passed down 3 and 4 generations, with only the first generation of the blonde diabetic mice having the supplemental diet with the B12 and folic acid. So doing something good passed down 3 and 4 generations, protecting the next 3 and 4 generations of offspring. Human studies, Netherlands during World War II was a severe food shortage. Children born of mothers, and the average caloric intake during this time, they were getting about 500 calories a day, which is a very low caloric intake. And the children born to mothers during World War II in Netherlands had higher rates of diabetes, obesity, and metabolic problems than their siblings of the same biologic parents. And they did studies in their genetics, and they found that they had 5% less methyl groups on a particular gene, and that gene helped the body absorb energy from food. And so with less methyl groups, it means it had a 5% increase in the expression of this gene, which means that these children could extract more energy from food. And so when mom's pregnant and you've got 500 calories a day, what's happening is the message is being sent, you're coming out into a world with very little nutritional resources for you to survive. So in order to help you adapt, we're going to decrease the methyl groups on this gene so that your GI system will be able to absorb more nutrients than your brothers and sisters who don't have this gene. And then, of course, the war's over. They get plenty of nutrition. They can eat the full, robust meal. But now they're still extracting more energy from the food, and they end up obese and diabetic with metabolic problems. A methionine-rich diet worsens memory. Mice, this was an animal diet, this is not a human study, but mice were fed a diet high in methionine, that's fish, eggs, and meat, and low in B12 and folic acid, and they had a particular gene that codes for a protein called netrin, methylated. More methyl groups were attached, and methyl groups shut it down, and resulting in worse memory. Netrin is a brain protein that helps synaptics, plasticity, growth of new neurons, axonal guidance, and making those connections between neurons. And so this particular protein being shut down this process of making new pathways and new neurons in your brain. When they introduced netrin, just intracerebrally with a syringe, they just injected some in there. It reversed 50% of the memory problem because they had this protein now available again. Mother's intake of mercury in fish oil. Study of 400 children over 8 years found that children of mothers who had a mercury level of 1 microgram per gram had higher rates of ADHD than children of mothers who did not have that mercury level. So mercury, and that probably doesn't shock anyone that mercury is not good for the brain. And then children of mothers who ate fish, though, twice per week had lower rates of ADHD. Because omega-3 fatty acids from fish are very important for neuronal membrane integrity and health, and they're anti-inflammatory. And omega-3s from most plant sources the brain can't use. Those are the short-chain omega-3s, the ALA omega-3, and the brain uses DHA, EPA, long-chain omega-3s, and that comes from fish oil mostly. And so the high fish oil omega-3s actually are very healthy for brain development. Additionally, studies show that women who are low in omega-3s have higher rates of postpartum depression because when they're pregnant, the brain is taking omega-3s from them in order to make a new brain, and omega-3s make women vulnerable to mood disorders and depression. And so I don't know how it is in Australia, but in America, essentially all the prenatal vitamins now are fortified with omega-3s in addition to some of the other vitamins. RNA in food. MicroRNAs, the little short scripts of RNA, act as signaling molecules altering gene expression, in other words, epigenetic modifiers. Plant RNA from food. When you eat food, you not only absorb the proteins and the fats and the sugars in the food, you're absorbing the DNA in the food. And some of this DNA gets expressed in your body, altering how your DNA is being expressed. It's very fascinating research. So yes, it's not just the molecules becoming building blocks. The DNA information can alter how your DNA is expressed. And in this particular study, this microRNA, which is abundant in rice, was highly enriched in the serum. In other words, when you drew serum of Chinese people who eat a lot of rice, you could actually measure this particular RNA molecule, which is from the rice. And this particular RNA molecule in mice and in humans altered the LDL protein receptor adapter, altering how your liver expresses low-density lipoprotein cholesterol. So brain circuits, parental environmental influences. So are you getting a sense so far that it does matter? See, a lot of kids you talk to go, this is my body, it doesn't matter, I'll do with what I want. Only if you're not planning on having children yourself. If you're planning on having children yourself, then what you're doing is not only affecting you, it's affecting your kids, your grandkids, your great-grandkids, and most likely your great-great-grandkids are all being affected by what you're doing with yourself today. What's the Bible say about that? Passes down 3 and 4 generations, doesn't it? This is exactly what the science shows. So brain circuits, for those who weren't here this morning, I'll just run through this really quick. Metaphor of fire alarm in the school building. You pull the little red box, set off the alarm. That's analogous to your amygdala in your brain. It's your alarm. When the alarm goes, it alerts your 000 operator, which is your emergency operator, and that is your hypothalamus in your brain. And the job of the emergency operator is to call in emergency responders, which come out of your adrenal glands, adrenaline and glucocorticoid. When the emergency responders arrive at the school, the scene of the fire, there's a fire chief who assesses how big the blaze is, how many responders we have, calls back to the emergency operator and says, hey, we've got enough, you don't need to send anymore. In our brain, those are hippocampal neurons. They register the rise of those stress hormones. They'll signal back to the hypothalamus and say, hey, we've got enough, you don't need to send anymore. And then there's a principal, an administrator, who assesses whether some kid's playing a prank or whether it's a real fire, and that, of course, is your dorsolateral prefrontal cortex. And so when the alarm fires, the dorsolateral prefrontal cortex will evaluate and calm down if there's actually not a threat there, as we talked about this morning. And then the salience network we talked about this morning, all sensory input as it's coming into your brain passes through your thalamus on its way to its ultimate cortical destination. If it's threatening to you in some way, it will activate your amygdala, your alarm circuitry, will tag that information, say, this is important, watch out, pay attention to this, and send this information up here for you to take an action plan. So, you all hear this fan blowing in here. Everybody hear it? It's been blowing the whole time. That activating your tympanic membrane, your acoustic nerve, going through your thalamus, but it's non-threatening. Your automated salience network said it's nothing, ignore it, and you haven't paid any attention to it. But if you smell smoke like the building's on fire smoke right now, your salience network will automatically say, pay attention, that's important. And so if this becomes disrupted, though, then the energy from the alarm circuitry, if it's disrupted at the anterior cortex level, doesn't come up here to be processed reasonably. It gets sent into your orbital cortex, and your orbital cortex's job is to give you a conviction of wrongdoing and redirect you away from doing inappropriate things. So as we talked about this morning, I know there's a few people that weren't here this morning, so I'm going to give these examples again to help them kind of catch up with us. And if you were to stand up in this room and try to take your clothes off in front of the rest of us, your orbital cortex would begin firing like crazy and activate your stress circuitry, and don't do it, don't do it. That's the job of the orbital cortex. And then, so if you disrupt the salience network, what happens then is this information that should be processed up here gets sent into the orbital cortex, but you get a conviction of guilt or wrongdoing, which activates more stress circuitry, and you get negative ruminating loops going. And the classic example is somebody who's lost a loved one, and they, in denial, no, I'm not going to deal with it. So they're not processing it here, so the energy goes here, and they'll go, it's my fault. If I would have called them and kept them on the phone 10 minutes longer, they wouldn't have been in the wreck. If I would have booked them on a different plane, if I would have done this, if I would have done that, and they get these negative ruminating loops going. And if the amygdala doesn't calm, it activates your sympathetic nervous system, which activates macrophages, which activates your immune system, which kicks up inflammatory cytokines, which damage the fire chief, damage insulin receptors, cause increasing glucose in your system, increasing the risk of diabetes, hyperlipidemia, obesity, strokes, heart attacks, all because of this disrupted neural circuitry. Now, what I didn't share with you this morning, what I'm going to share with you now, is what actually happens. So what I shared with you this morning, and we just went through really briefly, is if you have negative thought patterns, you disrupt your salience network, you activate the fear circuitry, which activates your immune system, causing inflammatory cascades in your body. Now I'm going to take you to the next step, how that inflammatory cascade reacts back on your brain. In order to understand, this is a synapse. This is a micrograph of a synapse. This is your presynaptic, and this is your postsynaptic neuron. This is these little vesicles here would be filled with things like dopamine and serotonin and norepinephrine. That dark area right there is the synaptic cleft between the two. This is just a drawing of that. The green is an astrocyte, And if you want to remember what an astrocyte does, think astronaut, even though it's spelled differently, and you'll remember, because an astronaut never goes out into space without their spacesuit. And the job of the spacesuit is to create a nice milieu or a nice environment to keep the astronaut healthy. The job of the astrocytes are to create a nice milieu for your neurons to keep your neurons healthy. They buffer potassium, calcium, make glutamate and other signalers, and they just create this nice milieu that your neurons like and keep them healthy. That's the job of the astrocyte. Now remember that as our next slide. Now this is your bloodstream, and you're stressed. You've got your negative circuitry going on. You've got your immune system kicked up. Your immune system's kicking out all these macrophages, which are kicking out those inflammatory cytokines, and now they're circulating your system. And there's another cell here that monitors your circulation called the microglia. Now the microglia normally will take a substance called tryptophan. Anybody ever heard of tryptophan? And do you know what tryptophan normally gets turned into in your brain? That's right, serotonin. It's the precursor to serotonin. And the microglia are one of the cells in your brain that make serotonin. Not the only, but one of them. And they will turn tryptophan into serotonin normally. Unfortunately, when this inflammatory cascade happens, this inflammatory cascade causes an epigenetic change in your microglia where the gene that is turning tryptophan into serotonin gets shut off, and another gene gets turned on, and tryptophan gets, instead of turning into serotonin, is now turned into quinolinic acid. Quinolinic acid is toxic to astroglia. And so you're out in space, this would be like having little micrometeors hitting your spacesuit and you start venting atmosphere. You are not a happy astronaut. Okay? And so what happens is with this inflammatory cascade, the astroglia get to be damaged, and the astroglia begin kicking out lots of glutamate, which open up calcium channels, and it rushes in, and this is toxic to neurons. We actually begin losing volume in hippocampal regions, the dorsolateral prefrontal cortex of people who have this type of thing going on, which is happening in depression, and you can actually see thinning of the brain in these areas. Additionally, remember we talked about BDNF this morning? brain-derived neurotrophic factor, fertilizer for your neurons. When it's there, the neurons will sprout new connections and so forth. This inflammatory cascade that I just went through in the neurons itself will cause an epigenetic modification where methyl groups will attach to the DNA region that codes for BDNF, and BDNF gets shut down, and you lose that protein, which keeps neurons robust and healthy. Interestingly enough, every antidepressant treatment that we've been able to identify that works, antidepressant medications, shock treatment, magnetic treatment, any of the treatments that work, epigenetically turn BDNF back on. And BDNF starts being produced, and neural sprouting starts going on, and neurogenesis starts happening again, and depression resolves. And with the medicines that turn on the BDNF gene, they do it not by removing the methyl group, but by attaching acetyl groups to the same region. And the acetyl groups basically pull the methyl group out, the shirt tail out of the zipper, basically. And they pull the methyl group out, and now the DNA can unzip and BDNF starts being produced again. The problem, I know all of you remember your organic chemistry. And the problem with organic chemistry in this is that acetyl groups have weaker chemical bonds, or another way to say that is methyl groups have stronger chemical bonds than the acetyl groups do. And so if somebody is depressed, they've had this whole inflammatory cascade go on, they come in and they've got their BDNF shut down with these methyl groups attached to their DNA that are shutting it down, and a doctor puts them on an antidepressant. And the antidepressant causes a second messenger cascade, causes the acetyl groups to go down into the DNA, attach to the DNA, pulling these methyl groups out, and the BDNF gets produced again. You get neural sprouting happening, neural regrowth happening. The person perks up, depression clears, they're feeling good. Wow, I feel so much better. Thank you, doc. Stop their medicine. Acetyl groups have weaker chemical bonds than methyl groups. The acetyl groups start breaking off. The methyl groups are still attached. BDNF gets shut down again. They start losing that neurotrophin protein. They start losing volume again, and they relapse back into depression. And every episode of depression, because every neuron, billions of neurons, have their own complement of DNA, and every neuron has the potential to have a methyl group attached to that DNA region. And so every time you're depressed, you get greater and greater saturation throughout the brain of this methyl shutdown. And it makes you more vulnerable to recurrence after depression is treated. So, animal studies, they took pups of nurturing mothers. So we've looked at pre-gestational, gestational, and now it's post-gestational, postpartum we're looking at now. So pups of nurturing mothers versus neglect moms. So these pups were born, and some of the pups had nurturing moms who would lick and groom them and pay lots of attention to them, and others had moms who just neglected them and wouldn't pay any attention to them. And they looked at brain development. And the pups with the attentive mothers had altered brain development such that they had, without the attentive mothers, the neglect mothers, had upregulated amygdalas, more anxiety, more stress. They were more socially impaired than those with attentive mothers. The point being is, mothers who attend, caress, love, hug, touch their children alter the developing brain such that they calm the fear circuitry down. Love is casting out fear neurobiologically. But if they neglect the child and they don't have a lot of this love, touch, affection thing going on, then the amygdala upregulates and they get more fear and anxiety. Neurobiologically, they'll be more anxious and worried and stressed kids. But some people thought, okay, hold on, maybe these neglect mothers are neglect mothers because they've got some genetic problem that causes them to be so stressed and anxious that they can't be good moms, and so maybe what you're seeing with this amygdala is the actual cause of the bad mothering, and it's not really, and they inherit this gene, and that's why they've got this upregulated amygdala. So they took the pups born of the neglect mothers at birth, and they put them in with these nurturing mothers who would lick and groom them, and then when they did that, their amygdalas developed normally. So it wasn't a genetic flaw. First week of postnatal life, rat pups were exposed to abusive mothering, and they had altered BDNF expression in their prefrontal cortex, and those rats grew up to be poor mothers. Their brains did not develop normally if they were abused in that first week. Their offspring were also born with altered BDNF and also had mothering problems, 2 generations worth now. If taken at birth from the poor mother and raised with a nurturing mother, there was some repair that happened and BDNF expression improved, but it didn't fully resolve the damage that was done in that first generation. It will take a couple generations of good mothering to resolve and undo this damage. Child abuse and gene expression in brain tissue, 41 Canadian men 25 were severely abused, and 16 controls. They looked at the DNA expression in the hippocampus, that's the fire chief area, and it's also, the hippocampus is where all new learning take place. If you're trying to remember what I'm saying right now, that's your hippocampus. That's like your RAM. It's your working memory. When you go to sleep tonight, memories get transferred out of your hippocampus into your cortex for long-term storage. If you use certain sleep aids, certain medications, they actually biologically in the hippocampus interfere with memory consolidation, and you lose data. So you learn something in school, you go take certain types of sleeping pills, and you sleep, and you can't get it into your cortex because the chemicals are interfering with the cortex transfer. So you lose data. All the Z drugs, basically, the zolpidem and the zolaploms, and I don't know what the brand names are in Australia, but most of those types of sleep drugs in the benzodiazepine category of medications do that. Anyway, they found in these particular group there were 362 different epigenetic changes from the abused group to the non-abused group, with some genes were hypermethylated, meaning shutting them down, some were hypomethylated, meaning overexpressing them, and most significantly were the genes that regulated neuroplasticity. The ability of the brain to rewire and restructure itself was impaired in people who've been abused. This was a very powerful study. It was actually done in New Zealand. It was a study over 32 years. They followed these people for 32 years. I wouldn't want to do a 32-year study. I like results faster than that. Anyway, in this study, they took individuals and they identified three measures of childhood adversity. One was overt physical or sexual abuse. That was one measure. Second measure, neglect, severe neglect. Third measure, severe socioeconomic deprivation or poverty. If you had none of those, you are represented in this blue bar. If you had one of those in childhood, you're represented in the green bar. If you had two or three of those, you're represented in the gray bar. And then they simply followed these individuals for the next 32 years and see who actually developed depression. And if you had none of those risk factors, 12% of people developed depression with none of those childhood risk factors. If you had one of those, 21% developed depression, and over 30% if you had two of those. And then they looked at metabolic problems such as diabetes, obesity, elevated cholesterol, these types of things, and you'll notice it parallels exactly the depression, 12%, 20%, 30%, depending on whether you're 0, 1, or 2. Because what I went through with you already today, I know you're putting the pieces of these puzzles together, because that childhood adversity is upregulating amygdala. Upregulating amygdala is causing those negative ruminating loops going, which is activating your immune system, causing an inflammatory cascade. And that inflammatory cascade is causing insulin resistance. And insulin resistance is going to damage your and the inflammatory cascade is going to damage your fire chief, which is going to call for more glucocorticoids. So you're going to get elevating glucocorticoids and insulin resistance, and you get this terrible inflammatory loop going. And that then reacts back upon the brain that I just went through, so you increase both depression and metabolic problems from childhood traumas and adversities. So adults who are abused as child have higher rates of medical illnesses, higher rates of mental illnesses, higher rates of suicide, higher rates of alcohol and drug problems than children who are not abused. Now, I've given you the negative side. Let's give some positive stuff. This was mice. This study was interesting. They engineered these mice to be stupid. They actually had a gene defect. They were designed to have a gene defect that caused memory problems, where they had trouble learning disability. These were special mice. And then they put these mice in an enriched environment for 2 weeks in their adolescence. And this is what a mouse-enriched environment looks like. They had extra toys and extra substances, extra smells to play with. And not surprising at all, the mice with the enriched environment had improved memory. They had more things to experience and go on. And so even with the gene defect, they had better memory than the clones that didn't have the enriched environment. But then they let this group of mice who had the enriched environment reproduce. And their offspring were born with improved memory, basically normal memory, despite having the gene defect and no enriched environment in their adolescence. What happened was the enriched environment in their parents caused an epigenetic modification that neutralized the gene defect such that even though the kids were born with the gene defect was not expressed and they had normal memory. And this modification happened at the level of the histones, where acetyl groups attached to the histones, which increased dendritic sprouting and connections, and they were able to overcome the gene defect because of the environmental experiences causing an epigenetic modification. Isn't that cool? Other protective factors for kids, family member confidant. If a child grows up in a home where they have someone they can confide in, they can actually share their heart and thoughts with, they actually grow up to be more adaptive, less mental health problems, less drug problems, less depression and so forth than kids without a family member confidant. And the other big protective factor is family cohesion, that sense that we have each other's back, that we watch out for you, that you can trust the people in your family not to gossip about you, not to tell your secrets, not to post pictures on Facebook that you wouldn't want posted, those types of things. You've got people at home that care about you and protect you. That is also helps kids have a downregulated amygdala so they grow up healthier, both physically and mentally. Other environmental factors: tobacco, alcohol, drugs. Tobacco, nicotine binds the receptors that regulate neural development. Teens who smoke have higher rates of psychosis, anxiety disorders, depression. They actually epigenetically alter the reward circuits, and they are more physiologically addicted if they start smoking before the age of 20 than if they start smoking after the age of 20. So their brain, it's more difficult to get off. Alcohol alters epigenetic. It's called neuropeptide Y, which is coming into a lot of research in PTSD individuals now, because the ability to express neuropeptide Y or not makes one vulnerable to having PTSD and trauma experiences. But anyway, alcohol alters the expression of this particular peptide such that when you're drinking, it causes an anti-anxiety effect during the intoxication, but then the gene expression changes, causing an upregulation, and then you actually get more anxiety afterwards. Marijuana, increased risk of psychotic disorders up to 40%. And we're talking this morning about the difference between God's law and man's law. This is a good example. I don't know what's happening here in Australia, but in America, a lot of states are making marijuana legal. Do y'all know that? Do y'all hear that in the news? Lots of states are making marijuana legal. It's a big, big debate thing going on. And see, states and human governments can pass laws to make marijuana legal. They can never pass laws to make marijuana healthy. You see the difference? This is the big difference between man's law and God's law. They can never do that. No matter what they say, marijuana is toxic to the brain. Damages white matter tracts, impairs prefrontal cortex function, lowers IQ for people who smoke before the age of 18. And young adults who smoke daily, they actually follow and look at the structure of the hippocampus in their brain, and they get actually physical structure changes that interfere with new learning and memory if they smoke daily. Media influences. Babies under the age of 1 watch approximately 1 hour of TV or more per day. At least in America. I don't know what's going on in Australia. This is from the U.S. 7 studies have confirmed that any television watching of any kind, including the old programs that are no longer available called Baby Einstein, delay language development if they watch television before the age of 2. American Academy of Pediatrics, American Academy of Child and Adolescent Psychiatry recommend 0 television of any kind under the age of 2 because it delays language development. So this program was marketed for a while, Make Your Baby Smart, show them all these videos and these programs, are off the market because it delays language development. Don't show them to small kids. There are no studies that show TV of educational material under age 2 is beneficial. Children get greater than 6 hours a day of electronic media of various kinds. 50% of teens in America have a TV in their bedroom. And the impact of television. Theatrical. Now we're talking theatrical television. We're not talking watching my DVD lectures, okay? That's not what we're talking about. That would be considered educational television. So theatrical television has as its primary effect to activate your limbic system emotion circuits while simultaneously turning off your prefrontal cortex reasoning thinking circuits. That's what it functionally wants to do to you. And if you remember normal brain development, if you don't use it, what happens? Yes, and so normal brain development, if you're firing something, it expands and gets stronger. If you're not, it's getting pruned back. Television watching wants to get you to laugh, wants to get you to cry, wants to get you afraid, wants to get you aroused, wants to get an emotional reaction while simultaneously turning off your thinking. And the stronger the emotional reaction, man, that was a good program. Okay, that's what we're looking for. For the first seminal study done on this, and it's been repeated in other groups, was done by Centerwall, published in the Journal of American Medical Association in '92. And he wanted to see if there's an association between television watching and violence in society. And he used a black and white indicator of violence, which is homicide rates in society. And he chose 3 societies, the U.S., Canada, and South Africa. United States and Canada, television came into those countries in 1945. No television was in the entire nation of South Africa until 1974. And so he wanted to look at homicide rates in these countries before and after the introduction of television. And then he used Canada as a control because Canada has very strict gun control laws. And if there was an increase in homicide rates in America, he didn't want people to say, yeah, but you got guns everywhere. So he used Canada. And in South Africa, he looked at white-on-white only homicide to take out the apartheid tensions that were going on during this time. And this is what he discovered. From 1945 to 1974, homicide rates in the U.S. increased 93%. In Canada, they increased 92%. And in South Africa, during the same time, they dropped 7%. And here's the kicker. He looked at South Africa from 1974 to 1987. After the introduction of television, white-on-white only homicide jumped 130%. Now, you might have heard preachers and other people talking about, well, if you put bad violent content in, you get bad violent content out, right? This modeling thing. This is primarily not modeling that we're dealing with in the sense of learned behavior based on behavior recognition patterns. We're talking altered neurobiology. Theatrical television alters the brain structure such by watching it, activating the limbic system mood circuits over and over again, you upregulate those mood circuits, you impair prefrontal cortex, which reasons, thinks, and self-restrains. I'll give you an example. You're on the interstate. As you're starting to get on the interstate, somebody cuts you off and almost hits you, and you get an irritable urge to say something ugly. That's your limbic system, that aggressive urge. But if you go, wait a second, I'm a Christian, I'm not going to act that way, I'm going to forgive them, and you restrain yourself, that's your prefrontal cortex. Television watching causes underdevelopment of prefrontal cortex, overdevelopment of limbic system circuits. So the more TV kids watch, by the time they hit adolescence and the hormones hit, then they have these raging emotions that they cannot process and restrain, so they act out aggressively, they act out sexually, or they turn to substances to try and calm themselves because they can't calm themselves, their brain can't do the work anymore. This is what's happening. It's neurobiological. Now, if you remember, do you know what programming was on in America between 1945 and 1974, the types of programs? I don't even know if you've even heard of these or seen these, but things like Leave It to Beaver. Anybody heard of that show? Lassie? Okay. These types of shows. And I could list a whole bunch of them. I Love Lucy. You've heard of these shows? Okay. All of them, every show is rated G. Every one from 1945 to 1974 is G-rated programming. G-rated programming, 92, 93% rise in homicides with G-rated programming. Now, the programming got worse after 74, and you get a magnifying effect. So if you get worse content, it'll magnify it, but even with G-rated programming, you get this problem. This was repeated in a somewhat different way. Zimmerman and Christaki in 2007 looked at three types of television. Violent R-rated theatrical television, nonviolent G-rated theatrical television, and educational television. And then they looked at measures of attention and focus and concentration, which is prefrontal cortex function. And what they found was theatrical programming, whether violent R-rated or G-rated nonviolent, impaired prefrontal cortex function and caused attention concentration problems. Educational programming did not. So it's not just letting your kids avoid the R-rated, it's also how much dosing they get of this medium because they're exercising emotion circuits while they're suspending their thinking circuits. That's the problem. The spiritual factors, we talked about God constructs in the brain. Newberg and his group, 65 years and older, they took individuals how to meditate 12 minutes a day on a God of love for 30 days, and at the end of the 30 days, they can measure growth in the anterior cingulate cortex where you experience empathy and love and compassion for others. They had lower heart rates and blood pressure because when this part of your brain is active and you're empathetic and compassionate towards others, this part of your brain actually turns off your amygdala, so perfect love is casting out fear neurobiologically. And they had 30% improvement in memory testing. So altruism, studies show that adults who volunteer, give of themselves after accounting for baseline measures of health, live longer, have less depression, less dementia, stay out of nursing homes longer, and stay independent longer than adults who don't volunteer. What about prayer? It's an interesting study. After September 11, 2001, the University of Michigan examined prayer and coping, and those who prayed daily had better outcomes, psychologically adjusted better 1 year later than those who did not pray. But then they also did another study on Muslim refugees of Kosovo and Bosnia, and they found that 60% of them had PTSD, and 77% of them had negative forms of prayer, such as praying their enemies would pay for what they have done. In other words, prayers of vengeance. And Muslims with positive prayers had high levels of optimism, hope, and healthy adjustment, but vengeance and anger-related prayers had reduced levels of optimism, hope, and healthy adjustment. So it's not just prayer, it's what you're praying, who you're praying to, that matters. Jesus said, you will know the truth, and the truth will set you free. I showed you this data this morning. If you do cognitive behavioral therapy, it actually normalizes the dysfunctional circuits and depression. How many didn't see this morning? Okay, there's enough in here. I'll run through this again really quick. Okay, they took individuals with depression and they compared them to controls, looking at activation of amygdala, your fear circuits, and dorsolateral prefrontal cortex before and after cognitive or truth-based therapy. They put them in the PET scanner and they gave them a mild stressor in the PET scanner to see what would happen to their fear circuitry. And the stressor they gave them is they showed them a picture of an ugly human face, and they said, well, that kind of looks like you, doesn't it? And that was their stressor. And the people with depression with that had significant activation of their fear circuitry. The control group with the dotted line did not. And then the depressed group after this truth-based therapy, cognitive therapy, had significant normalization of their amygdala response to that stress. And then they had them do a digit span, which is taking those numbers and put them in order from the lowest to the highest. And if you're doing that right now, you're activating your dorsolateral prefrontal cortex. And the non-depressed group did it quite nicely and smoothly. The depressed group struggled with it. And then the depressed group after cognitive therapy had more normalization of their dorsolateral prefrontal cortex. Why? Back to what I said earlier about normal brain development. If you exercise it, then it expands and gets stronger. And cognitive therapy is exercising in with the therapist, making the person think and reason and work through stuff in their dorsolateral prefrontal cortex. And as it exercises that, then it will actually calm the fear circuitry down, and their self-restraint is recovered in doing this process. So it's a brain exercise type therapy that will result in actual neuronal regrowth in healthy brain regions. Cognitive therapy and gene expression. This is epigenetic stuff we're talking about, right? PTSD patients had lower expression of a gene, of this particular gene, which resulted in, and so methyl groups attached, shut this gene down. This is your fire chief being shut down. And when your fire chief being shut down, your glucocorticoid receptors being shut down, you get smaller hippocampuses, you get thinning of the orbital cortex, so forth. Well, after cognitive behavioral therapy, PTSD patients didn't have a difference anymore in controls because this particular gene got turned back on. They actually started expressing their fire chief, which was holding their 911 operator in check again, and they had hippocampal regrowth. And so thinking new thoughts, the study shows, thinking new thoughts, healthier thoughts, alter how your genes are being expressed in your brain in healthy ways. Or saying another way, thinking negative, pessimistic, fear-based thinking will alter your genes in negative ways and damage your brain over time. Cognitive stress management therapy, 199 women with breast cancer randomized to either 10 weeks of cognitive behavioral stress management or active controls. And they looked at their white blood cells and they assessed their gene expression at baseline, 6 months, and 12 months later. Baseline negative effect associated with greater than 50% differential in these, basically there's 201 transcripts that were affected by more than 50% expression through this. But what was noted was there was upregulation if you had negative affect, upregulation in the pro-inflammatory genes and metastasis-related genes. So if a woman was stressed, she was pessimistic, she was negative, oh, I'm going to die, all this kind of negative stuff, it turned on genes that made it more likely she'd have metastasis. If she went through the cognitive stress management, however, 91 genes changed expression by more than 50%, specifically downregulating the pro-inflammatory genes and upregulating the type 1 interferon response genes. Those are your anti-cancer genes. So positive truth-based therapy and thinking healthy thoughts helped your genetics fight the cancer. How healthy spirituality helps reduce mental illness. Healthy spirituality is a spirituality that activates prefrontal cortex and anterior cingulate cortex, calms amygdala, slows and reduces the inflammatory cascade, thus improving mental and physical health. Altruism helps in the same way by activating anterior cingulate cortex, calming amygdala. With that, you have reduced anxiety and worry, so you have reduced ruminating loops. You have a healthier lifestyle oftentimes, so you have less oxidative stress in putting substances that are inflammatory into your body. You usually have healthy relationships and ability to forgive and not grudge hold, which helps stop the negative ruminating loops and reconnects your salience network. And so all of these things, healthy spirituality is actually physiologically healthy for you. So a healthy brain is optimized by Healthy parents. So pick healthy parents, please. And I know we can't pick our parents, but for those of you who are thinking about being parents or know people who are about to become parents, education can help them do things that can significantly benefit their children and grandchildren and so forth. Healthy intrauterine environment. So again, educate people you know to do things that are healthy, both mental, spiritual, physical things that are going to make a very nice milieu for the developing child. Proper nutrition for you and your brain still today. Affection, responsive parenting. So parents who are, if you have kids, really be sure that you're able to nurture, spend time, cuddle, hold those children. Lots of skin-to-skin touching and so forth. It's very good for brain development. Physical activity. This is something, I don't know how the school systems are here in Australia. In America, the public school systems are just really in trouble. They're so the federal government is so into their business that they can't actually do what they think is good for the kids, and they have these requirements that they have to meet now with the 3 R's, reading, writing, and arithmetic. And so a lot of things are getting cut out of the school systems that used to be there, including a lot of physical activity. And we know now the way your brain develops that the neural circuits that are involved in motor movement and initiating motor movement, which are dysfunctional in a disease, tell me what disease? Parkinson's disease, that's right. So people with Parkinson's disease lose these neurons that initiate motor movement, and so they have a hard time initiating motor movement. And if you know somebody with Parkinson's, if they're not on their medications, they also have a hard time initiating thought. They have a hard time expressing themselves because the same neurons that initiate motor movement initiate thinking. And then that's the initiation of thought. And then the part of the brain, the cerebellum in the back of your brain, helps you coordinate smooth motor movement and balance and things. And so when you're riding your bicycle, your cerebellum is involved. And in any type of motor coordination that you're doing, your cerebellum's involved. And we now know that the vermis of the cerebellum, which is the central portion of the cerebellum, helps coordinate and make your thinking smooth and coordinated. So we want kids to get out and play. We want them to develop their physical skills. We want them to be able to coordinate their bodies because in doing this, they're developing the very circuits they're going to need to coordinate their thinking. But much of the public education in America doesn't do this anymore. We want to stimulate the 5 senses, music, art, and things like this. Develop those circuits as well. Avoid toxic substances. I have one patient who struggles with addictions, and he tells me when he was a kid, his mother put whiskey in his baby bottle to calm him down from crying. So let's avoid that stuff, moms, okay? I know you've got to work on that. Education. Yes, education is very important, and we do want to do the cognitive education, but we don't want to lose the practical education just to learn the cognitive education. So we want to hands-on education too, and teach kids how to actually do stuff. Teach kids how to be responsible. Chores around the home, really good for kids. Can you believe I just said that? Limit theatrical education. Limit theatrical education. Entertainment. Limit theatrical entertainment. And you notice I didn't say no at theatrical entertainment because in Western society it's really virtually impossible not for them to get some theatrical entertainment, but limit it. And if you're going to have some, you could also do some things that would engage their thinking in the process, like when you're going to have a movie, if you're going to do that, say, hey, afterwards we're going to have a little discussion. What did you learn from that? What was the point of that? What was the metaphor? This movie is a metaphor of what? How does this connect to the great story, the great theme? Do you know most great stories are actually just spin-off of the Great Controversy? That's what they are. Most of all of the great stories are spin-offs of the Great Controversy. You can see it, Matrix. If you look at Matrix, this is the Messiah. You look at Harry Potter, dying and rising saviors. I mean, you see it. All these metaphors are there. But so have a dialogue and discussion. Make them think about it. Healthy spirituality and altruistic endeavors, we want to include that. Resolve guilt and forgive other people. Don't hold grudges. Very toxic to do that. Sleep, I didn't go into sleep, but sleep is one of the... so I see some adolescents and young people in here, like adolescents meaning from age like 13 to 25, you're an adolescent because your brain doesn't finish developing until you're 25. And these college students, young college students and high school students don't understand that there are 4 physical requirements to have a healthy brain and to be alive. Air, water, food, and sleep. Sleep is a physical requirement your brain has to have. And if you actually don't get any sleep, you can die from not getting sleep. So I have to educate to get your sleep because your brain, the first part of the brain that gets impaired when you're sleep deprived is prefrontal cortex. So there's an old wives' tale that goes something like this: don't go to bed angry with your spouse. You ever heard that wives' tale? Toss it. What I say to my wife, and she can verify this, is if we have a discussion that we need to get into that's of any substance, it's not something real simple like where'd you put the toothpaste, okay? But something of some substance, I'll say, I love you too much to give you only half of my brain. You need my entire brain. So let's sleep on this and talk about it tomorrow. And then you'll find if you do this, when you wake up the next day, your prefrontal cortex is on, your irritability level's way lower, your tolerance level for frustration is way higher, your ability to restrain impulses is much better, so you're much less likely to say things that you don't mean that cause things just to blow up. And those late night dialogues are the worst time to get into it with your spouse. So, because your prefrontal cortex is being impaired. So summary, brain is highly complex and susceptible to change. Environment causes marked changes in brain development, and environment alters gene expression, which is passed down through the generations.